have bacteria that are cocci, bacilli, and then there's one that we didn't say a whole lot about, but sporilla or spirochetes. That's kind of like another name for it. I think syphilis or treponema pallidum, and we're going to talk about one that causes Lyme's disease called Borrelia burgdorferi. That's another spirochete that you'll see. That's the two that just come to my mind immediately when I think of that. There are, remember that organism that causes syphilis? It's called treponema pallidum. And I think that's in black bold on your PowerPoint if you're looking at it. Treponema pallidum. In case before I mention this, I just wanted to make sure and let you guys know, we're fixing to talk all about syphilis, and it can end up being a horrible disease and all of that, but your basic medication or treatment for syphilis is penicillin, right? It is a very easy fix if they go to the doctor.
If you're allergic to penicillin.
Well, then they have to give you another type of antibiotic. That's a good question. That's the first thing I thought. Well, what about the poor people that are allergic? You know, that's the first thing they ask you, are you allergic to penicillin? But there are other antibiotics they can use in place for those people that are allergic to it.
So does the antibiotic kill it, or do they always have syphilis?
No, the antibiotic kills it.
Okay.
Now, they say that it can come back because syphilis, even, as we're gonna talk about, it can come back in the form of the herpes and all of that type of stuff. It all is based back to the virus that causes that, yeah.
So your folks that are gonna be allergic to penicillin, that's a type 2 allergy, isn't it?
Being allergic to certain drugs actually can fall over different ones of those hypersensitivities that we talked about. All I can tell you is that I won't do that to you on the test. I wouldn't ask you a question to where there's 3 answers. But like if I said, for example, transfusion reactions, that's type 2. Okay? Or if I said peanut allergies, that would be type 1, right? Okay. If I said mycobacterium tuberculosis, that would be type 4. Remember, I gave TB as an example.
Oh yeah, because I wrote that down, but I've slept.
All right, there's 4 stages of syphilis. You have a primary stage, secondary stage, third stage is the latent stage, where basically the symptoms and stuff go away, but right after that, you can fall into the tertiary stage. Some books kind of put the latent and the tertiary stage together, but it's not going to be together for this class, all right? So 4 stages: primary, secondary, latent, and tertiary. It says untreated people can transmit the infection during the primary and secondary phases. An infected mother can pass this on to the fetus. This is called congenital syphilis. The fetus is most at risk in the 4th month of gestation, all right? Now, as soon as I read that for the first time, I remembered it because we used to have to do an automatic, when I worked at I keep calling it St. Joe because I'm old, all right? But when I worked there, every baby born, the cord blood sample had to have an RPR test on it. It was a state mandated test. Basically, that is checking to see if that baby may have gotten syphilis because it was passed to the baby from the mother, all right? That used to be state mandated. I don't know if it still is, but that used to be a state mandated test. So the mother can pass that on to the baby. Now, I'm pretty sure that anytime a mom goes in for a prenatal workup or anything like that, I'm pretty sure that's common protocol to test the mom for syphilis. Now, they probably won't say a syphilis test. It'll be an RPR or a test called a BDRL, which we're going to talk about a little bit, okay? But an RPR, that is a test that tests for syphilis. All right, primary stage. This is just basically for your entertainment. I'm not going to, if I was to ask a question on the test, I'd probably have to tell you about the chancre. It says in the primary stage, the first symptom is a chancre. Usually appears within 2 to 6 weeks of exposure. Approximately a third of these untreated patients will progress on to chronic stages. Secondary syphilis, a skin rash will develop with brown sores on the palms and the soles of the feet. Appears 3 to 6 weeks after the chancre appears. This can cause a systemic infection. Symptoms are usually mild, with fatigue, headache, mild fever, sore throat, swollen lymph nodes. The symptoms may disappear without treatment, but may reoccur over the next 1 to 2 years. You may lapse into something called latent syphilis. Again, I'm thinking this is when you don't have treatment. This may be starting to come up, so I can get this on board. I've got something. The latent stage, patient is no longer contagious and is often asymptomatic. In the tertiary stage, the spirochetes will spread through the lymph system. They can lodge in the aorta, or cardiovascular lesions can develop. You can have an inflammatory response, which may continue for years. You've got a condition, neurosyphilis, that can occur. They check the spinal fluid for that, okay? When you take body fluids from me in the fall, we're gonna talk about, when we get into talking about spinal fluid, there is something called neurosyphilis that they test for. This is in the very last stage. It's never good to test positive for syphilis in the spinal fluid, okay? Because that means it's basically it's already in the brain, basically.
What makes them not contagious anymore?
You guys are asking me doctor questions. There's just certain, as diseases progress, they've just done studies to where there's certain times during the disease that they're more prone to pass that on to others. I mean, I'm not answering your question, tiptoeing around it, but that's just the way it is. I don't know. Bring about it. Haley?
Google.
No, I'm just joking. I don't know, that's something you can research on. All right, there's a good picture of a chancre, but I can't show it to you. It's probably black and white unless you printed yours off on
It's
color.
terrifying.
There's a chancre on someone's finger.
It's like necrotic tissue. Yeah.
Yeah, just a big mess.
I will tell you though, everything I read says those chancres, they usually don't, they develop on the first thing that touches an infected chancre, okay? So since this is transmitted sexually, even though the picture you're seeing is on the finger, I'm not trying to get gross, that's not usually where it first develops. Alright,
It's on the
it's
genitalia.
gonna develop on anything, let's think sexual characteristics, on the male genitals, the male penis, the vagina, the anal area, the rectum, okay? That's where the chancre usually develops first. When you guys, had a discussion with this, with Jason yesterday, when you take micro from Jason, he's got explicit pictures of chancres that you'll see, okay? So, just trust me, this picture is nice. I want to say that. So you can see the chancre on that person's finger, and then you can see the area of secondary syphilis where you start to develop a rash on the palms.
Normally they're spicular. It's like on the palms when it presents
like
like
they
in
need
circular
lotion.
patches.
It just looks like their hands are dirty.
It's very strange.
Dirty with syphilis.
Remember, guys, you're seeing horrible pictures and all that stuff, but penicillin can treat this. This used to actually be, not that uncommon, but it did go away, and now they say that it's coming back.
Oh, I have a fun fact about syphilis. I have a fun fact about syphilis.
Alright, what's that?
It was commonly found in llamas and was not discovered until after Columbus crossed to the New World.
It was found in lava?
Llamas.
Llamas, okay.
Yeah.
Llamas.
Lava, volcanoes,
I think
They used to treat people at the bathhouses, like Al Capone and stuff. Like when you walk through and tour them, they would have syphilis treatments in the hot springs, hot water, which didn't do anything for them, but they thought it was, or what have you, which I find to be crazy.
koalas also get syphilis.
Chlamydia.
Or chlamydia, that's what it is.
Watch out for that.
Yeah, that's why
Yeah.
we give them everything known to man, every vaccine, everything. That'd be okay.
But again, just don't lose sight of the fact, this is not anything like HIV or anything like that. The only way this gets to those stages is if someone just lets it go.
Right.
Okay, but the research that I did, even though I know that chancre you're looking at on the finger looks obvious, what I was looking at was saying that sometimes these chancres, for example, if they, I'm not trying to talk bad, but like if the chancre is very light, and say it gets under the foreskin of the penis, so it gets into the deep inside the vagina or
Oh, they
something.
can't see it.
It's not, you can't see it.
Right.
And so then it can progress on to, later stages.
Wow.
Alright, so now that we get out of the nursing and the doctor stage of it, let's talk about what we do, the testing of it, okay? There is 2 tests, that you can do for syphilis, and you need to pay attention here. There's 2 types of tests. There's non-treponemal tests, and there's treponemal tests. The ones that I'm familiar with is the non-treponemal, and this is called a VDRL, and then there's another one that's called an RPR. That VDRL stands for Venereal Disease Research Laboratory Testing, all right? That is one of the older tests that they used to do. If we ever had a VDRL where I worked at, we sent that one out, but if you go to National Park, and I think even now, I don't know if CHI is doing it or they're sending it out, but it's an RPR test. And we are going to do that test today in lab, because it'll go along with this lecture. We will do the RPR test. It's a little bit more hands-on and a little bit more manual, all right? It is based off of, well, they really don't call this agglutination, they call it flocculation. And what flocculation is I want you to kind of think more along the lines of a soluble, all right? It's actually going to be using an antigen, and you are trying to detect an antibody that we're going to talk about that is a reagent antibody. But it is, a reagent antibody is not an antibody that develops from treponemal pallidum organism. It is an antibody that basically is produced from the damage that that organism does to the body. Okay, it does damage, and then there's an antibody that basically is produced from the damage. I want you to think of it that way, to that antibody. It's called a reagent antibody. Hence the word RPR. All right, so let's read the treponemal test. We'll talk a little bit about that. There's one called a fluorescence treponemal antibody absorbance test, and then there's an MHA/TP, all right? That's a microhemagglutination for treponemal pallidum. But if you have to study 2, I want you to study the VDR and the RPR, because you will be faced with that more than you will be with others. Okay, I'll read about them to you, but I hope those are more important. Okay, the VDRL test. This is a classic serological procedure. It's a flocculation test in which an affected patient produces reagent antibodies. These antibodies are against components of their own infected cells. The test uses an antigen made up of cardiolipin, cholesterol, and lecithin. The reagent antibody is not specific for syphilis, and it can also be detected in other infectious diseases like TB, measles, hepatitis, mono, malaria, etc., even in pregnancy. You can do the test in CSF, that's spinal fluid, for that disease I was telling you about called neurosyphilis. False positives can be seen in other inflammatory conditions. One of the reasons that a lot of people, they don't like this test is you have to inactivate complement in the serum before you perform this test, and it has to be read underneath the microscope. The test that we're going to do is the RPR. You don't have to inactivate the complement, and you don't have to have a microscope. You can visually see it on a little ring like the test that you guys have been doing, all right? But there's just a little bit more to it. So the RPR test. This is a slide test that uses modified VDRL antigen. The antigen uses charcoal and a dye for visualization. It can be used in the field. There's no microscope. You don't have to inactivate complement. When you guys open this kit today, you're going to have a bottle in there. It looks like clear fluid, but on the very bottom of it, unless Jason pre-mixes it for you guys, when you mix it up, it's going to look like a charcoal solution. It's going to be a real dark color. This is the antigen you use, okay, to detect the reagent antibody in the patient's serum or plasma. Make sense? Okay, so you'll use an antigen that, I call it charcoal, but you do kind of need to know the ingredients that's made of, the lecithin and all of that. Okay, the treponemal methods. Just going to read about these. The first one is the FTA absorbance, or the fluorescent treponemal antibody absorption. Basically, the patient's serum is first absorbed with a non-treponeme and palatal-treponemal antigen to reduce the cross-reactivity. The serum is then overlaid onto the slide, fixed with the treponemal antigen, and then a fluorescent anti-human antibody is added. So I'm glad I didn't erase that, because that's pretty much that picture right there. That's what they're talking about. You have a glass slide, all right? And then you are actually trying to find the treponemal antibody, and you'll see a form of fluorescence. I've never seen that test done. The microhemagglutination treponemal pallidum uses treated red blood cells coated with treponemal antigens. The presence of the antibody causes the red cells to agglutination in a microdilution well. Now there is a way, and I think this is important, look down just underneath that, under the note. There is a way that you can directly observe the spirochetes. It says if you use a certain procedure called dark field microscopy, you can visualize the modal treponeme spirochete itself. You do this by performing a wet mount. All right, if the patient has moist lesions, I'm assuming by this they mean the chancre, okay? They can go into the chancre, and I just want you to pretend like, okay, so maybe it's wet and moist. They can get a drop of fluid out of that. They can put it on a microscope slide, and there's a way that they have a dark field microscope that they can actually see the organism that's causing it. Okay, that would be a definite positive result if you see that. All these other ways that we're testing, we're not testing directly for the organism. We're testing because of something that was produced because of the organism, right? Like, it's really hard to see the hepatitis virus itself. We have to see the antibody that's produced because that virus is in that antibody. Viruses can be very, very small. Sometimes you need an electron microscope to see them. I've never even seen an electron microscope. They're usually only found in major college research, like say a Duke University that's doing research. They may cost $1 million just for the scope itself. I think in your intro to MLT book, if I remember, you got some pictures that show you electron photographs of things like what a platelet truly looks like in its natural state, and a red blood cell in its natural state. Any questions about that? All right, next page, you should have a picture of, it looks like a mechanical rocker or a mechanical rotator. Do you guys see that? We'll be using that today. When you do an RPR test or a VDRL test, the instructions tell you, now up till now, we've been letting you guys hold the cards and look for agglutination, like in the D-dimer, those glass cards. For whatever reason, for this particular procedure, you have to use a mechanical rotator. You even have to calibrate it. You're supposed to check it so often and make sure that it makes a complete circle so many times within a 1 minute time frame, right? Because when you check this for agglutination, after you add the charcoal and the control or the plasma, it's supposed to rotate. It's going to tell us, I think it's 7 to 8 minutes, all right? And you do this one a little different. Depending upon what kit you're using, you actually lay down your plasma and you fill in that circle, right? That circle well, like I've always been telling you guys, take up as much of the circle as you can without going outside the lines. Like you're learning how to color, right? Stay in the lines. But then when you add that charcoal, you don't mix the charcoal. You add 1 drop, a perfect drop, right on the top of it, and you just let it lay there, and then you let the rotator mix it in, okay? You don't actually take a stirrer unless we're using a kit that is different from anything that I've ever used, and it has to go for 8 minutes, and then you take it out. These are not going to be, they're not going to just look like boom, agglutination like you guys are used to seeing. You really got to look at these. They actually have controls that are called a negative, a weakly reactive, a moderately reactive, and a strongly reactive. And again, it's not really agglutination you're seeing. It's more like flocculation, where you see that little bit of a black charcoal, and you're looking for making sure there's not a tail going around with it. The best way to do it is just for you guys to do it, and you'll see what I'm talking about when you look at it. All right, the febrile agglutinin test. You guys are gonna like what I'm fixing to tell you about this test. It's very outdated. I haven't seen this test performed in forever, so we are going to whittle this down, and I'm gonna tell you what I want you to know about febrile agglutinins, okay? The first thing, I just wanna explain why they don't use this test anymore. This is a test, there are so many different types of bacteria that you can get. There's things like Proteus, Salmonella, there's all these different types of bacterial infections you can get. And there used to be this test called a febrile agglutinin, and I used to hate when I would see this order, because you would have to get, you know that little black card and the 3 rings you guys have been doing? Imagine one of those where the circles are smaller, and it's a plate about 12 inches by 12 inches, and there's like 9 circles going across, 9 circles going across. And what you basically have to do is they made a special pipette where you would have to make a serial dilution of your patient's serum on each line, like go 1 to 1, 1 to 2, 1 to 4, 1 to 8, 1 to 16, all right? And then you add the corresponding organism that you're looking for as antigen. But it was kind of like a batch test where you were testing for 3 different strains of Proteus, 2 different strains of Salmonella. I'm not so sure. I think they had some that were called OX. And I didn't work in micro, but all these different organisms. But it took forever to do this test because of all the dilutions you had to make. So basically, you had reagents that had all of these different organisms' antigens in it. You had to make a dilution going all the way across of your patient's serum to see if they possibly had antibodies to those antigens, all right? If they did have an antibody, you had to report out not a positive or a negative, you had to report out how far it went out as far as a titer, like what dilution was it positive to, okay? And by that, I mean, for example, let's just say that we were testing for Proteus, that's a certain organism. So this well had, again, I think it went out 9, but I'm only gonna draw 6 here just to get my point across. This was your straight solution, okay? So obviously, it was positive, okay? You had the Proteus antibody in the plasma because you had the antigen, there's the agglutination, right? This was the 1 to 2 dilution that you had to make. And so of course, you know, you might see a little bit of agglutination in that one, maybe a little bit of agglutination in that one. So you would have to report out the febrile agglutinin for the Proteus antibody was positive up to a straight 1 to 2, 1 to 4 dilution, right? But physicians had a cutoff point to determine if it was significant or not, all right? Like if it went out past a 1 to 20 or a 1 to 40, that would be a positive result, depending upon which antigen you were testing for, okay? But again, this entire line would be testing for the Proteus antigen. The next 3 lines might be testing for Salmonella, different species of Salmonella antigens. You see what I mean? Anyway, it was just a very manual test. Even someone that did it all the time, if you set up a febrile agglutinin, it would take you at least 20 minutes, okay? There should be no test that takes take 20 minutes to do. That's too long, okay? So that's the nuts and the bolts of the febrile agglutinin. So you're welcome to read over that. Some of the other antigens that I'm looking at on the PowerPoint, Brucella, that's another one that they look for. There's an organism called Francisella tularensis that causes tularemia. That's another one that you had to look for antibodies for that one as well. So let's move to page 4 on your PowerPoint to talk about serological testing for today. Guys, this computer's up, but we don't really have that much further to go before we're finished with the material, so let's just keep rolling with this, okay? Serological testing for group A strep. We did this yesterday. We're testing for group A beta hemolytic strep. Now, a lot of the stuff that we're talking about today, and even in serology period, you guys are gonna hear this stuff again and probably go in more detail when you take microbiology in the fall, okay? Serology is based on a lot of the tests that we do in serology are to detect microorganisms, but we're wanting to detect them quick. In other words, we don't have time to wait for that agar plate to grow out the organism. That takes 24 hours. So they've developed all of these antigen antibody reactions to detect all of this type of bacteria, okay? So like I told you guys yesterday about strep, we're fixing to talk about, a lot of times they'll get 2 swabs on a person. One of the swabs is for the quick test that they can determine. How long did it take you guys to do that strep test yesterday? 5 minutes, something like that? Probably quicker if you've done it a couple of times, 5 minutes. So if they can get a presumptive positive on a serological test, they can go ahead and start that patient on antibiotics, even though they're taking another swab and they're inoculating an agar plate and they're gonna check it for growth the next morning. If they have a difference in results, they'll call that patient at home, see what I mean, and go, hold it, there's a problem, all right? But more than likely, they know if there's gonna be growth or no growth, or if they're gonna find that group A beta hemolytic strep on that plate the next day. Okay, the name of that organism that they have to worry about in strep is called Streptococcus pyogenes, all right? That's a group A beta hemolytic strep that can cause a lot of problems. It says it can cause a wide variety of infections. The most common are gonna be acute pharyngitis. It can cause that URI is upper respiratory infections. Impetigo, that's some sort of a bacterial infection on the skin. It could go on to cause scarlet fever, problems with the eyes, otitis, pneumonia, glomerulonephritis. We're gonna learn all about that in your analysis, glomerulonephritis. But basically, if someone gets group A beta hemolytic strep, that actually eventually can go away and heal itself, but it can cause other systemic problems. So they've got to fix that. They've got to get rid of that as quick as possible, because that can progress to worse diseases. The rapid strep test that you did yesterday, first marketed around 1980. You need a throat swab to do that test. You get immediate results instead of having to wait 24 to 48 hours for the throat culture results to come back. Still remember, and it's in parentheses there, the throat culture is still the gold standard, all right? It's growing it on an agar plate and then testing those colonies to see if one of those, how they can determine if that organism is beta hemolytic is they will streak that on the blood agar plate, and when they hold that blood agar plate to the light, it's like it ferments, or it basically, I call it, it needs that blood and that blood agar plate to grow, and you'll see a clearing behind it. In other words, the red will be clear where you can almost see through it.
Is that why they put an A-disc on the blood, for you to be able to tell if it's hemolytic or not?
A-disc is, no, an A-disc contains an antibiotic called bacitracin, and they're looking to see if that organism can grow up to the disc or if that organism is inhibited by the disc. That's something you guys will learn in micro, but it's a neat concept. I'll explain it briefly just so you kind of understand how important it is. If we have an organism that grows on an agar plate, like let's take a blood agar plate for example, and we swab somebody's throat and we wipe it on that blood agar plate and we wipe it all over it, okay? We can place antibiotics on that agar plate, okay? The antibiotics, they're little paper discs that contain so much amount of an antibiotic. So we may put 7 or 8 different antibiotics on there, right? And we'll place it on that media. When that organism grows, you can tell which antibiotics work because the organism won't grow close to that little paper disc. So that's how we tell the physician, hey, you need to use this antibiotic because this antibiotic is not gonna work for this infection because the organism grew all the way up to it. That means that antibiotic has no effect on whatever's causing you to be sick. But if you see a big zone around that little paper disc, that means that antibiotic don't want no part of that. The organism doesn't want any part of that antibiotic and it is inhibiting it. It is causing it to stay away. That's how they know what antibiotic to give a patient. But they can't find out that information until about 48 hours after they did that initial swab, all right? Okay, the reaction and the rapid test is considered an enzyme immunoassay procedure where you have a colored product. Again, it determines the kit you're using. Most kits use an enzyme immunoassay procedure. The sensitivity of the rapid test is not sufficient to permit their use in pediatric populations without an accompanying throat culture. Best approach to follow up a negative rapid with a throat culture. So even if that test is negative, even if you've got that 7-year-old kid that they're worried about, do they have strep throat? Even if the rapid test is negative, they are still gonna take the second swab that they used, they're gonna streak it on a blood agar plate, they're gonna check that plate the next morning about 24 hours and see if they had any growth, and then a microbiologist can just look at the plate and they'll look for certain colonies that are beta hemolytic, and then they will do a test on it to see if that's group A strep, right? In certain situations, if you don't have a strong infection, the rapid test, the test you did yesterday, can be negative, but you can still have a little bit of group A beta hemolytic strep grow out on that agar plate. That's when that parent's gonna get a call the next morning and says, look, we know we sent you home and said, don't worry about it, but we called you in a prescription for an antibiotic because your son or daughter does have group A beta hemolytic strep, okay? So have you guys ever had to go to a clinic where they swabbed your throat with 2 swabs and not just one? Well, that's the reason for the second swab, if you ever run into that situation.
Why sucks.
All right, another test we're gonna do today is called the ASO test, the anti-streptolysin O. Strep, group A beta hemolytic strep, produces a toxin called streptolysin, all right? Streptolysin O, the patient will build an antibody to streptolysin. This is called an anti-streptolysin O antibody. It says the original ASO test used red blood cells to detect the lysis. Now latex agglutination has largely replaced it. That's what you'll be doing today, is a latex agglutination test for the streptolysin antibody, all right? This is just another quick way to determine if someone has got that pathologic bacteria in their throat. It does also say that molecular techniques are being designed as well. This can consist of PCR, DNA probes, etc. Question?
And when we do this test, and we're in search of the antibody, that means that the test will come equipped with the antigen in it.
Yes.
Thank you.
It has the actual streptolysin. That's what's coated on the beads, is streptolysin, and it's trying to see if your patient has the streptolysin antibody, anti-streptolysin. All right, moving on to the next test. We're gonna do this one today. This will be your third test, okay? All the tests we're gonna do today hopefully is gonna correlate with this lecture, or that's the way we planned it. We're gonna do a test for mononucleosis. So just a little bit of recall. What is one thing we can see on a differential that can indicate that a patient may have infectious mononucleosis? Reactive lymphocytes. Don't forget that. Somebody described to me basically, what does a reactive lymphocyte look like?
Smash between the red blood cells.
Bumped up against red blood cells.
And it has the outer lining of the blue. It's like darker on the edges.
Yeah, good. And you were talking about the red cells. They're gonna basically push in on the cytoplasm of that lymphocyte. It distorts the lymphocyte, and it makes it hard to actually sometimes determine, is it truly a reactive lymph? Remember, there were some that were debatable that I told you, I don't know if it's a monocyte or a lymphocyte, but I go by a couple features. If red cells are pushing it in and I see that dark bluing color on the very remnants or the edges of the cytoplasm, I tend to go lymphocyte, right? If they can still do it to monocytes too, and I don't know what to tell you other than experience and time. After you look at so many, you can just tell on that. All right, mononucleosis is caused by a virus called the Epstein-Barr virus. This is a human herpes virus. It says it's usually benign. It's a self-limiting lymphoproliferative condition. Usually has a 4 to 7 week incubation. Affects the B lymphocytes of young adults and children. Symptoms can vary. Patients will be tired, have a sore throat, fever, cervical lymphadenopathy. Basically, that means your lymph nodes swell up. and your spleen enlarges. I've had that before, and I've had a bad case of it, and I know I have to go to the doctor every week, and they're always checking your lymph nodes, because your lymph nodes swell up in your neck. What they're worried about is your spleen. Your spleen really gets large, and you cannot take a direct hit there. Like when I actually had it, I used to play football, no football, couldn't play football for a year, no contact sports of any kind, because your spleen does not go back down to original size for quite a while, okay? So, there's different severities of mono. Some people get a light case of it, whereas some people will get it really, really bad. But that's one thing they have to watch out for, is your spleen enlarges, and you can't take a hit to the spleen. So they'll probably, a physician, he or she's gonna tell you, no contact sports of any kind. So for those of you that play rugby, anything like that, if you ever get mono, they're not gonna let you do that. Epstein-Barr virus causes it. Even though it's not on this PowerPoint, I mean, I can't really test you on it, but remember those reactive lymphocytes, because that's important for mono as well. There can be cross reactions or complications. They mention other disease states that it can progress into. They talk about Bell's palsy, Guillain-Barré syndrome. Have any of you ever known anyone that's had Bell's palsy?
Yes, my mother.
That's a bad condition. We had an instructor get it, Paul, who's next door, and he had it for like 5 months. Half of his face was like
It
paralyzed.
just doesn't work.
Yeah,
It's
'cause when he-
strange.
Because when he
Yeah, and there's
came to
nothing
talk
you
to our
can do
class
about it.
Nothing.
It basically just causes partial paralysis. It's gotta get better on its own. But I'll tell you another condition that's worse than that one is that Guillain-Barré. I had a relative that had that, and that's another thing that they're not really sure why, but it's like the body, all the organs just like stop working. They usually have to put you on a vent. I had a relative that had that Guillain-Barré. It's rare, but she was in the ICU for probably 3 months. And I mean, they basically said she was gonna die. I mean, because she was on a vent, body organs weren't working, having to use a feeding tube and all this stuff. She made a 100% recovery. She's as normal as all of us in this room right now. But I'm telling you, everybody, was going to visit her, because the people were saying, look, that condition is basically, it's really, really bad and you may not recover, but if you do make it through it, a lot of people have a
Just
full
fine.
recovery. So if you guys wanna read about that.
It says here it's a complication from mono. Did your relative have the mono?
Not that I know of, no one ever knew. There were doctors coming from all over the state to look at her case, just because they were, it was just like, boom, just a normal person, like, you guys go home tonight, and then tomorrow you're in ICU. It is a complication, they think, for mono, but I'm sure there's other things that it can be a complication from as well. All right, heterophile antibody. You do need to be aware of this. I'm on page 5 of your PowerPoint. It says antibodies that are stimulated by one antigen but can react with antigens of other mammal species. What I want you to know about a heterophile antibody is that is the principle of how we determine if someone has mono by these kit tests, all right? We are detecting a heterophile antibody. That antibody can react with antigens from animals' red cells, like cattle, sheep, all right? All of these other things. Now, they're gonna make it complicated and say bovine antigens and all this different stuff, but I just want you to understand, somebody that has mono, that develops an antibody, that antibody is called a heterophile antibody. And one thing that distinguishes it from other types of antibodies is they have figured out that a heterophile antibody, people that have mono, will react with certain animals' red cells. And when you read over this, it's going to tell you one of the red cells is guinea pig kidney. All right? Have I ever done this? No. Okay, but I'm just telling you about how these reagents occur. All right, so read over this, but don't lose sight. It gets a little complicated, but what I want you to know is people that have mono develop a heterophile antibody, and that a heterophile antibody will react with antigens from animals, certain animals. Okay. Now, the next PowerPoint that has this Davidson differential test, Don't study that, okay? I don't think you're gonna need to know that, all right? So just, you can cross that out. I just want you to know that they calculated, they determined which animals the heterophile antibody will react with, but I believe if I remember right, there is no reactivity in guinea pig kidney red blood cells. Just about all of the other animals, those antibodies will agglutinate with red cells from different animals, but they've determined the guinea pig kidney, it won't react with those red blood cells, right? Last one we're gonna talk about, and we'll take a break before the review, we're gonna talk about Lyme's disease. We live in Hot Springs, Arkansas, all right? We live in a rural community. I'm sure all of you know what a tick is, okay? So that means you've probably at least heard of Lyme's disease before. It is caused from an organism called Borrelia burgdorferi, and that is found in certain ticks, okay? The host for this organism is the deer, all right? So that's how it's basically can be transmitted to humans, but this is a very, this causes people to be very, very sick, and it's also hard to diagnose because I've had relatives that actually have had this condition, and they were sick for 6 months, and they never could figure it out, and after they finally recovered, they said, well, maybe it was Lyme's disease.
That's my mom got that Rocky tick fever.
Rocky Mountain spotted tick fever.
Sure, they thought it was cancer, and then they took them long before they actually diagnosed her with it.
Well, and it seems like Lyme's disease and that go hand in hand as
That's
far
what
as
it was
being
really
hard to diagnose. Okay, so it says it's transmitted by, again, that hard-bodied tick. The host is the white-tailed deer. It says it emerged disease of last 25 years, virtually unknown in the U.S. until 1969. It has been reported in all 50 states, difficult to diagnose. The tick bites a human. There will be spirochetes that migrate out from the bite in a unique expanding skin lesion. It says it has a bullseye rash. I've never seen that rash, but that picture does look like a bullseye. It says if left untreated, can have multi-system involvement of joints, heart, nervous system, symptoms of headache, rash, flu-like symptoms. The treatment is certain oral antibiotics. We don't have one this year because we used it all up, but we did have a kit for Borrelia burgdorferi that we could order from Standby that tests for Lyme's disease. It's a simple agglutination kit. One drop and one droplet for agglutination, and it actually can detect the antibody caused from that organism. It says antibodies in this condition are usually not detectable until symptoms have been present for at least 2 to 4 weeks. It can be chronic if left untreated, can develop into arthritis. You can detect it, antibodies using different methods. ELISA is one of the ones, and then again, they're doing research and figuring out how to use molecular techniques like PCR and DNA. It does say false negatives are due to slow rise of antibody, false positive if the patient has been exposed to bacteria that are similar in nature. I would underline the part that says Lyme's disease is caused by organism Borrelia burgdorferi. That's, again, we're just touching the surface on this stuff. You will talk a lot more about these conditions in microbiology.